beneath this nice green layer of vegetation there is a soil which in some places can go to a metre or more. Here in West Wales the soils tend to be fairly shallow, maybe 10-20 centimetres or thereabouts, and it's really vital to understand the important processes that happen here. The dead bodies of all the plants that are growing here are decomposed in the soil, and the process of decomposition is vital for the carbon cycle of the planet, because the carbon dioxide that's fixed by the plants is then released in decomposition, and it's particularly the fungi in the soil that are really important for this job. The other thing that the fungi also do is, having decomposed the plant material, they feed back the nutrients, the nitrogen, the phosphorus and the other elements back to the plants. They link up to the roots of plants in structures called mycorrhizas, and it's a particular group of fungi that form mycorrhizas with the plants that I'm particularly interested in, because they're very sensitive to many of the things that we do in modern land management and agriculture, particularly ploughing and fertilizer addition. These fungi aren't used to these disturbances, and they're essentially destroyed from the habitat when these things happen, and it can take them decades, possibly centuries, to come back in. In this part of the country, this lowland area, much of it has been ploughed. So I've brought you here today to Plas Llanecheiron. It's an 18th century manor house built by the famous architect John Nash. We're not really here to look at the house itself, it's the lawn that is particularly interesting to us, because the lawn is probably the same age as the house, and it's been managed just by being clipped ever since the house was built. And therefore these sort of ancestral, these older fungal populations still exist here, and we know that because if we come here in the autumn we see this huge diversity of colours of these waxcap fungi and various other types of fungi like fairy clubs. But today I'm here in the height of summer. The rain here might make you doubt my words on that point, but this is not really mushroom season, and what I'm going to do is I'm going to sample the soil. The main implement I'm going to use today is a soil corer. I can push this into the soil, maybe only about 10 centimetres this far. I can turn it a little bit and I can extract the soil core. I'm not just going to take one soil core, I'm going to take maybe 30 or so from this particular area, because I want to average out over the whole area, and I want to pick up all the fungi. The fungi have their little domains. I can then mix all those cores together, and when we come to extracting the DNA later on, we can see which species are present and how abundant they are in that particular soil sample. Here's one of the cores I've just taken. It's compressed a little bit from the 10 centimetres, but you can see I've deliberately captured some of the vegetation on top. So the roots of the plants are there, and if the fungi are inside the roots, we'll find them, as well as the fungi in the soil. And you'll notice that despite it being rather wet weather past week or so, this part is quite dry. And it's important to appreciate that the structure of the soil as well as its composition are really important. All these little channels that all the soil animals have made are allowing air in, and they allow water to shed off the surface and to drain evenly. It's important for the function of the soil that oxygen can get in, and that water, when it's dry, can also get in, but not too much of it. Because the fungi, whilst we see them in the autumn with the mushrooms being formed, and we can appreciate them then, they're around all year. In fact, they live probably longer than we do, just gradually growing, degrading the soil organic matter, feeding it back to the plants. So when you see a mushroom, you have to remember that's just the tip of an iceberg, and the business end of the organism is in the soil. And in every metre squared of soil here, there are sort of a few hundred grams of waxcap mycelium just working away, turning over the organic matter, just gradually, and feeding those nutrients that are remaining back to the plants. Because of this really poor summer weather we've had, it's fooling all the fungi into fruiting. And here we have the persistent waxcap, this is called, Hygrocybe persistens. It's made a mushroom here and also here, just a matter of centimetres away from the edge of the house, and the wall is just here. This one is quite an interesting one because it is known to fruit quite early. You tend to find it in late August, early September, as opposed to sort of the October, November time that most of these waxcaps usually fruit. So this is an example of one that might be missed if we did sampling in the autumn. You'd have to come back now and also in the autumn, whereas when we sample the soil DNA, we capture all these organisms because they're all there in equal abundance all year round. As well as collecting DNA from the soil, we need something to match up the soil DNA to. So I'm going to pick this one carefully, I'm going to dry it, and we'll take a little piece of the cap tissue and we'll extract DNA from that and get its DNA sequence. Okay, so I've taken my soil cores. You can see there's maybe 20 or so from this little area of the lawn. The next step is to take them back to the lab and process them so we can get the DNA out of them. But now that I've taken these organisms out of their natural environment, things can start to happen in there that might change the fungal community. So I'm going to make sure that I keep the samples cold in these ice cube boxes in this insulated box, just for the travel back to Aberystwyth and to our lab where we can process them further. Okay, so we're back in Aberystwyth now at the Aberystwyth University, and I have my samples here. Still feel they're nicely cooled, not frozen, so biological activity has been slowed down there, but I want to get them frozen as quickly as possible in this ultra low freezer. So in a couple of hours those soil samples will be solidly frozen, and then they'll be ready for freeze drying, where we remove the water from the sample directly to vapour, so that there isn't any water present and biological activity is stopped. And then we have a dried sample without any degradation of DNA or the mycelium of the fungi. Okay, so now we've freeze dried our samples, and they've been in this desiccator here. So this is, they're warming up to room temperature, but making sure that they don't become damp again. And you can see from the bag, I can feel this is slightly cold to the touch, but it's bone dry, and I can crush it. It makes it very friable. You can see behind me now, my colleague Andrew Detheridge is grinding the soil through a fine sieve, a little bit like this. You can see he's wearing gloves to avoid cross-contaminating different samples, and using a pestle. So the idea here is to grind the stuff into small particles and to mix it all up, so that maybe 500 grams of DNA of the soil that we had originally is then ground up and mixed up very efficiently, so that when we take a small subsample, less than 1 gram from that amount, it's representative of what I sampled earlier. So we're now moving from the dirty lab where we ground the soil to the clean lab, the molecular biology lab, and we're going to extract the DNA from the soil. The first thing to note is that we use a remarkably small amount of DNA. This tube is only maybe 1/5 full of soil, about 1/5 of a gram is what we use for this procedure, and the kit we use is called Power Soil. It's designed to extract all that mucky humic material away from the DNA, so that all the molecular biology procedures that we use later will work properly and not be inhibited by the mixture of stuff in soil. And the first step that we have with this procedure is to mix the soil with these beads in a buffer, and that will bash, the glass beads will bash against the soil particles and break open all the fungal hyphae, and they'll release their DNA. This is the first step of the DNA procedure. My colleague James here is just in the middle of one of these procedures, which maybe takes for 10 samples a couple of hours to do, but then we have pure high-grade DNA for the later procedures that is representative of what we found this morning in the field. We've purified the DNA, and we've also amplified the DNA barcode region of the DNA, and this is us looking at some samples to see that by and large they've nearly all worked. Some have worked better than others, and you can see there's a brighter band of DNA here than others. And the next step before we sequence all this DNA is to make sure we have the same amount of each one in each sample. So here we are at the next generation sequencer. This particular one is an ion torrent personal genome machine. It wasn't designed to help mycologists look at waxcap populations. It was designed to sequence human genomes, specifically to be able to pinpoint mutations in cancer, for instance. So that was the driver for making this type of technology. The heart of this technology is this wonderful little chip here. On this chip we can get 5 million DNA sequences from this. It has 10 million little holes in it. In each hole a little bead coated with DNA will fall and will be sequenced, mostly to do with the release of acidity. The ion torrent runs overnight, and then it takes a few hours for the vast amount of data that is generated from one run to be crunched And basically the crunching involves matching up these sequences with reference sequences, sequences that have names. Because as a mycologist, that's what I want to know, is how many species are there, what are the species, and how abundant are they? So here we have a list. This is a typical data set, including some of the samples that we've just looked at. And here you see in pink are the waxcaps and the fairy clubs. And many of the top 20 most abundant organisms, or most abundant fungi at this site were either waxcaps or fairy clubs. And I notice in particular we've got most abundant of all is the red Hygoscybicoccinia. That's a very pretty bright red mushroom. About 10th down here we have the pink waxcap, Porpulomopsis calyptriformis. Also present is the meadow waxcap, Cucuphilus pratensis. That's pretty abundant. The abundances are shown here. So you can see in blue more than 5% of all the fungi in a given sample. So these things are also pretty. They're the film stars of the mushroom world, but they're also playing an important role in these soils. And our work will continue to try and work out exactly what they do. More than 500 years ago, Leonardo da Vinci said that we know more about the celestial bodies than the earth beneath our feet. It's pretty much true now, but as you can see, this technology that is starting to be developed by us and for other organisms by other people is going to start to bring soil to life, to tell us who the organisms that are important in soil biology are. And it's not just the waxcaps, which are the ones that have a bit of charisma and colour about them, but there's thousands of species in this hugely biodiverse ecosystem that we need to understand in order to be able to understand how soil functions and be able to preserve our soils.